just review that Costas is a professor of neurosurgery who is also a true scholar. As you know, he's an MD PhD with his PhD from University of Pittsburgh, and he's been an international leader in developing fluorescence-guided surgery for malignant brain tumors, and is in that rare cadre of physician scientists who, while operating, has been able to get enough preliminary data in his lab to successfully put it all together into closing the deal on his R01 as PI. So it puts him in very, very rare company. And he's also a really nice guy and great colleague. So Costas, we'll give it to you to introduce the talk today.
Okay, thank you, Dr. Betterson. Hopefully you guys can hear me. I'm at South Nassau today. So this is really a treat for us all. I think I am going to give a talk actually first, and then Dr. Ivkov, who you see here, will give the second talk. This is a really tight collaboration that we've had for a number of years. Dr. Ivkov's at Johns Hopkins. He's an associate professor in radiation oncology, and he's really been just a tour de force in the cancer nanotechnology world, specifically focusing on magnetic nanoparticles. And that's really what our research has been based on together, I think now for the last 4 years. I'm going to get in, when I get in my talk, I'm going to add something more about that. But, you know, I think Rob has really been a thoughtful leader with magnetic nanotechnology and its application to cancer in particular. And he's done work with prostate cancer, breast cancer, and I think pancreatic cancer. So he's really well versed in this field, and it's been great working with him as a clinician scientist. It's always important to surround yourself with people who are smarter than you. And I think, you know, Rob is one of those individuals who just kind of helps us think of new ideas and ways to apply the research. Okay, so can I share my screen now, and then I'll show you my presentation?
Yep, you're ready to go.
Okay, so. All right, so let's put this in presentation mode. So I have no disclosures for this. These are my other disclosures with 5-ALA, as Dr. Betterson mentioned, and Synaptive, none related to this. So I show this slide here because this is an interesting slide from 2011. It was a Gordon Research Conference, in Colby College up in Maine. And this is where I initially met Rob. And the reason why this is, I feel like, a symbolic slide is, you know, this is a conference, the first Cancer Nanotechnology Gordon Research Conference. Somehow I got invited to this as the lone surgeon there. And it was just, I felt like I was a kid in a candy store. It was a bunch of hot, really well-known scientists, researchers, and they were just all together in a conference room. And I was just able to interact with so many people. And Piotr Grodzinski he was the NCI, the National Cancer Institute Director of Cancer Nanotechnology. And he went on to lead the Centers of Cancer Nanotechnology, which were big U grants that went out 4 or 5 years later. But Rob was there, and that's where we had a first chance to interact with each other. But this is a great example of both intra and interdisciplinary science for our residents. I mean, I think that's where some of the real translational and ideas can be born that really can have an impact and be meaningful. This was my talk back then, and it was really on just a whole different concept, but we had developed these nanoparticles with an antibody, cetuximab, and we were using those to target GBM, and you know that it took off from there. But this has been a project that's been going on for a number of years, and it's been modified and tweaked. So here's the agenda I'm going to talk about. This is about glioblastoma treatments and challenges, magnetic nanoparticle theranostics. We'll talk about convection enhanced delivery with these nanoparticles, and then really jump into the magnetic hyperthermia therapy concept and paradigm of glioblastoma, and then talk about some of our models that we've developed at Mount Sinai. So everybody on this call, for the most part, knows that we always try to do a safe maximal resection for glioblastoma. And then these patients go on to other adjuvant therapies, and at recurrence, of course, we try to re-resect if possible, or enroll in a clinical trial, or re-irradiate. But, the problem is these tumors just keep coming back. And, while they don't metastasize to outside areas from the brain as much, very rare, they do locally invade and recur. And it's just very challenging to resect every cancer cell. You can never do that. And part of that is the biology of the cancer. It just infiltrates into the surrounding brain. And that's been known for decades. We focused on magnetic nanoparticles because of their theranostic strength. So they can be used as imaging agents. And in fact, they are administered IV for contrast now. But the other aspect of them is in their multifunctional nature is that you can tag them with things. And you can also load them with things such as siRNA, drugs. And then the other neat concept is what's called local hyperthermia from alternating magnetic fields. So we're going to get to that right now. But this is kind of a summary slide that shows how multifunctional these nanoparticle agents are. And part of this I was alerted to by my dad, who's a physicist. It was literally a dinner conversation years ago. And my dad's a magnetics researcher. And he and I would always just talk about, how can we apply this to brain tumors? And, I knew that the strength of these nanoparticles. My dad was involved with developing some of the newer class magnets. And, the whole concept of heat in cancer is not a new concept, but how can we, use magnetic nanoparticles to generate heat and kill cancer cells in a controlled local manner? So that was something that, kind of was born out of those dinner conversations and then kind of took off from there. But you can see here, you can target the cancer cells with antibodies. You can heat them up with an alternating magnetic field, which oscillates the nanoparticles and generates heat. And then you can image them by MRI scan. You can see here this rodent MRI scan of a GBM where we delivered the nanoparticles. They're hypointense. So they're multifunctional, and each of these have their strengths. As I mentioned earlier, our early work involved tagging these nanoparticles with cetuximab so we can bind to EGFR-V3 on GBM, which is a very characteristic mutation of these tumors. And we're able to do some publications with that new work. And then, of course, convection-enhanced delivery was another mode of delivery that we could bypass systemic delivery with all its inherent difficulties, and something that's near and dear to most neurosurgeons because we can implant catheters and really robustly deliver agents that normally couldn't get to the brain because of the blood-brain barrier. And we know recently with some of the work out of Columbia and others that you can deliver CED chronically. I mean, you could do this over days. You can put subcutaneous pumps in and just continuously pump whatever you want into the brain safely with multiple catheters. Some of our early work with nanoparticles involved using a canine model at the University of Georgia. And these were just some of the really, really early MRI scans that we used where we could see the agent we delivered in the brain. And that was a big deal because at the time, convection enhanced delivery, you just can never see what the agent was doing. And then later, groups would start co-infusing gadolinium so you could visualize that. But still, you wouldn't see the actual agent itself, which is important to understand how it distributes and if you're targeting the tumor. So these are just some of the work. So we noticed these things hang around for weeks in the dog, but they were safe. They were non-toxic at the time. And in fact, we were able to treat some dogs with these targeted nanoparticles that we designed in our lab in Emory. This was Petey. Petey became a famous pit bull down south. He had a very large glioma that was partially resected. We implanted catheters and then infused the cetuximab nanoparticle construct over 3 days, and his tumor actually disappeared. This was another dog in that same study. You could just see the catheter and the nanoparticles being delivered into and around the tumor with the hypointensity as you see here. This was Petey's tumor. It was just a large glioma. And, the canine neurosurgeons just biopsy, for the most part, these tumors. And you could see the nanoparticles really were able to target the tumor. And he lived for 2 years, but then developed a recurrence. And he actually died because of his hip dysplasia, which just became so bad. Laser interstitial thermal therapy, I'm going to switch gears a little bit, is one of the areas that we actively use hyperthermia for right now to treat brain tumors. And that's something that we do at Mount Sinai. We did a case on Monday where we implant a catheter into the tumor with stereotactic guidance, and then we use MRI to really understand how to heat the tumor up and target it. And this has really paved the way for a number of treatments for recurrent high-grade gliomas, recurrent brain metastases, and really helping us understand how hyperthermia can treat brain tumors. Well, the other thing that we've learned is that the blood-brain barrier is disrupted by LITT. And there's been a recent paper from Albert Kim's lab at WashU where they showed that the BBB can be opened up for at least 30 days after LITT. So that's been kind of a neat concept to explore, because if we can use heat to open up the BBB, then we can maybe get things into the brain tumor that we normally couldn't, and potentially target those resistant tumors. So hyperthermia and GBM is something that's been reported. The Europeans, as always, have beat us to the punch, but they described, their studies with hyperthermia and glioblastoma years ago, and what they found were that between 41 and 44, you could inhibit cancer growth. So you don't need really extreme hyperthermia. Just moderate hyperthermia can induce apoptosis of GBM cells. And it can also enhance the effects of radiation and chemotherapy. Radiation through double-strand breaks and other types of mechanistic enhancement. And then, of course, with chemotherapy, disruption of the blood-brain barrier. Heat shock proteins are elevated with local hyperthermia, and that may generate an immune response. So the Germans were able to get European approval for magnetic hyperthermia therapy with a very rudimentary nanoparticle construct. And basically the concept was you would stereotactically inject this into the tumor, and then patients would undergo multiple sessions of alternating magnetic fields. These are very safe. They're not heat or anything like that. They're just magnetic fields that oscillate, and they basically vibrate the nanoparticles. And when you have enough nanoparticles that are magnetic enough, then you generate a local heat effect. And they were able to show better patient outcomes when they combined this with radiation therapy. It was published in 2011. And it just never took off because of the concentration they had to deliver into patients, the contraindication of MRI scan afterwards because of the increased amount of metal in the brain. So we've kind of picked up from there and moved on. And just for everyone to understand, the mechanistic details of magnetic hyperthermia therapy are currently being studied, but there's a number of mechanisms where tumor cell death occurs. So not only by shear force from the oscillation, but you can induce apoptosis, you can, promote more DNA double-strand breaks, and all sorts of things with chemosensitivity and other types of processes. So that's something that we're actually actively studying with the tumor microenvironment, and it's opened up just a whole new area. So we talked about the limitations of the existing platform and the high concentration, and then you have to inject the nanoparticles into tumors, which probably results in backflow and not really proper targeting. And no one's really looked at combining chemotherapy and radiation therapy, and that's something that we just published on in a review recently. So here's the concept here with the illustration: is you would deliver these nanoparticles in and around the tumor, and then control the heat within the tumor with treatment planning, almost like radiation planning that Rob is going to touch on in his presentation, and really focus the effects and heating effects on the tumor. This is the model we've developed at Mount Sinai. This is the alternating magnetic machine that we use in our rodents. You can see actually a rodent on a cooling blanket, and the head's placed between this coil. And we have temperature probes in the brain and also in the rectum, so we can really measure the temperature effects. And this is something that we've been studying and doing some groundwork to understand how well these nanoparticles can heat in the tumor. So we're not opening the skull, we're implanting the nanoparticles and then coming back later and putting the whole mouse head in there with the coil and administering the field. So we're not going to get into the physics of all this, but we have different parameters that we use for alternating magnetic fields. It's measured by Gauss and kilohertz. And that's something that we can actually adjust in our treatments. So our initial work was with the Johns Hopkins University nanoparticles that Rob's group had developed. And these are just some heating waves here that you can see comparing to other types of nanoparticles, including the FDA-approved Faradex. And you can see Faradex, which is used for anemia, it's a magnetic nanoparticle, it's just weakly magnetic and hyperthermic, whereas the JHU nanoparticles, which we used with our initial work, were quite impressive in their heating effect when exposed to a magnetic alternating magnetic field. And see, this is some of our early work that we did with the JHU nanoparticles. And I want to point out a few things here. So the red curve is the heating curve when we apply the magnetic field ipsilaterally. So the blue curve and the black curve are not the area where the nanoparticles were deposited. The blue is the contralateral brain, and the black is the rectum. And you can see right when you turn on the magnetic field, that red line clearly separates from the other lines. And then when you turn the alternating magnetic field off, then that heating effect dissipates. It's very, very impressive. It's almost like a switch on, switch off. And it's very selective to that area. So we've switched to using a new generation of nanoparticles that we've collaborated with a German company, and Rob's been helping us through this. And these require a 20-fold lower concentration, and they really heat well. And these are probably going to be taken for FDA approval for MRI contrast enhancement in patients. So they're very soluble in water. It's remarkable what they've done with the surface chemistry of these nanoparticles. We haven't even talked about that. That's a whole nother area and talk, is how you make these things biocompatible. So this is some of the work that we've done recently. And you can see here, the heating effect is pretty pronounced. I mean, we're getting over 50 degrees Celsius in the ipsilateral brain. And this is a volume dependent effect. You can see here with just 5 microliters, 6, 8, and 10 microliters, how high things can go. And in the rodent brain with the alternating magnetic field. And, you can see right when you turn on the AMF, that field and temperature, the temperature immediately kicks in. And here we can go over 60 degrees Celsius. So this is really, really promising to do this in a rodent model, because now we can test all sorts of things in addition to radiation and chemotherapy combinations. We've done a number of volume-based studies. We've done a number of concentration-based studies, and we've found a very similar effect here with this pronounced heating effect at the site of the nanoparticles. The other thing that was kind of interesting too is that when we apply the magnetic hyperthermia therapy 3 days after we implant the nanoparticles, we still have this heating effect. So the question is, why is that important? Well, clinically, if we can just deposit these nanoparticles in our patients, and then we could basically put them through treatments, days or weeks later, that becomes a neat treatment paradigm, because patients could have their nanoparticles implanted and then, undergo multiple therapies with the alternating magnetic fields to help kill their tumor. I almost envision a patient going to an AMF room, and then afterwards going to the radiation room right next door. So they would kind of get their AMF treatment, then get their radiation treatment, or vice versa. And we're actually studying whether one impacts the other. This is another thing I want to show you here. So with our model, we're able to actually maintain the temperature at a certain range. So this is a rodent we treated, and we maintained the hyperthermia effect around 45. So the machine can turn on the AMF field on and off just to maintain that temperature, so you can make sure that we're not burning the brain, in other words. So we can pick any temperature we want. We know we can go up into 50 or 60 degrees Celsius, and we can really just kind of keep that temperature in a somewhat stable range, 1 or 2 degrees, plus or minus 1 to 2 degrees Celsius. So, we're doing toxicity studies, of course, to make sure they're not toxic. They do cause seizures, so we've had to give our rodents Dilantin. About 25% develop seizures. Once we preload them with Dilantin, there's really no issue with the nanoparticles. So that's been something we've learned. And all our GBM patients are on antiepileptics anyways. I don't think that's anything that would be different from standard of care. Here are some other studies that we've done combining the treatment with radiation therapy. These were the original JHU, Johns Hopkins University nanoparticles. And you can see here with bioluminescence imaging that we really can knock down the the signal from the tumors and create an anti-tumor effect when we combine the MHT with radiation therapy. So this is one of our early studies really showing the combined approach is very helpful. And then we also wanted to understand how well temozolomide penetrates the xenograft with or without hyperthermia. In this experiment here, we took xenografts and the surrounding brain tissue out. And you could see here on the right, the green with magnetic hyperthermia therapy, the temozolomide concentration is much higher with the heat. And that coincides with the other work done at WashU and others with increased opening of the blood-brain barrier with the heat application with LITT. So this is the newer version with MHT. There's a whole area that we're studying with treatment planning, and Rob will touch on this. And this is a very important concept because we want to control the heating of the tumor and limit the heating of the surrounding brain. And that's where some of the tricks from treatment planning are going to be important. And part of our grant is designing that so we can translate that to humans. We've done some preliminary survival studies combining radiation, chemotherapy, and MHT. This is one of our most tumorigenic models. It's a V3 expressing xenograft. These mice die between 12 and 13 days after implantation. And we only use 20% the dose of TMZ here just to see if we detected an effect. So there's definitely something going on here, and this is our worst player in regards to models. So we're studying this in multiple other, both rodent GBM models as well as human orthotopic and PDX models. In our grant, we propose using some rabbit models to also do some treatment planning experiments. This has kind of changed now. One of the PIs ended up leaving Hopkins and went to the NCI to be a program officer, but this is something that we're still interested in. And this is a sarcoma rabbit model that really looks just like GBM and invades the brain like the GBM. So it's a model that we're still kind of looking at to study our applications. But we're actually going to use the laser interstitial thermal therapy data as well to develop treatment planning, because again, we're using heat in that platform as well. And we have continuous thermometry done by MRI scans, so we can collect that data, understand how the heat dissipates in and around the tumor. Well, I'm just going to wrap it up here at the end here. So we have a canine feasibility study for glioma patients there that we're gearing up to do. COVID's really put a wrench and monkey into all this stuff, but we've been slowly planning this, and we're about to launch it. And we're still kind of waiting to see what happens with COVID, but we are going to have canine glioma patients that we will implant catheters, do CED, and do the MHT treatments in combination with radiation and chemotherapy. So I'm going to conclude here. I think Rob's going to take over after here, but I just want to just introduce this to our department, just to understand some of the novel translational work we're doing. These are the magnetic nanoparticles. Really are something we've learned a tremendous amount from. I think delivering them well, understanding the alternating magnetic field parameters that we could generate for heat, and combining them with the adjuvant therapies, I think is going to be important as we take this to the next level. So I think the canine glioma model is really going to help us translate this to humans in a phase 1. And that's really our ultimate goal here, is how we can get these to humans. And the nice thing is the nanoparticles can be produced in a GMP-like manner from Europe. So I think we'll be good to go once we develop some of our safety data in our preclinical models. So can't do any of this without my entire lab. Want to thank Alex Burris and our entire lab who've done so much work with this. And of course, prior collaborations at Georgia, and then our new collaborations with Johns Hopkins. And Penn State's actually part of this grant as well. They're going to help us compile some of that LITT data. So thank you. I'm going to stop sharing. Maybe we can wait till the end for questions, Peter, or it's up to you guys. Yeah.
I think that's fine.
Yeah. So Rob, are you able to get on? Are you ready, Rob?
Good morning. Yes, I am ready. Thanks for the introduction. I am a little reluctant to speak now because after that wonderful introduction, I think I'm only going to basically just prove you wrong.
I don't think so.
Okay, so let's see. I will go ahead and share my screen. Did that work out well?
Yes.
let's see, I'm trying to put it into presentation mode, but my computer is not cooperating to go into presentation mode. There we go. Okay.
That's perfect.
All right, so I am going to thank you again for the opportunity, everyone, to speak with you. And Costas, thank you for the wonderful introduction. I will basically pick up a little bit more, elaborate on some of the points that Costas talked about. And I will elaborate a little bit more on some of the other applications, and really specifically on some of the other features or aspects of magnetic nanoparticles. My disclosures, I do consult and I'm on a scientific advisory board for a couple of companies that are developing magnetic nanoparticles and also magnetic particle imaging technology, but none of that is part of this talk. Acknowledging collaborators through the years. So the work that I will be talking about is actually looking back and not looking forward, or not really part of the current project that Costas talked about, but this is the background on the materials. And over the years, I've been fortunate enough to scrape together some funding along the way. And to manage to basically develop and keep working on this, what I think is rather an exciting clinically translational technology. So some of the material that Costas already talked about is going to really be sort of the beginning point, just to get everybody on the same page. So really the motivation for magnetic nanoparticle or magnetic fluid hyperthermia really stems from the advantages of thermal therapy. And that is, in a very high level, that heat or heat stress enhances the effects of DNA damaging agents such as ionizing radiation and some chemotherapies for cancer through complementary mechanisms. To my knowledge, there is no thermal therapy, hyperthermia therapy, that is indicated for use as a standalone treatment. It's really, hyperthermia is indicated as an adjunct to radiation therapy or chemotherapy for recurrent cancers. And so that's sort of where I see the space for the hyperthermia. And this is unlike ablation or ablative therapies, which are essentially for the most part perisurgical techniques and can be used more or less as standalone. And you guys know much more about that than I do. And as Costas mentioned, the effect of combining heat and ionizing radiation is really to inhibit, because the heat inhibits DNA damage repair, primarily because it denatures or induces conformational changes and therefore inactivation in various proteins, enzymes. Some of these are transcription factors that are required for DNA damage repair. But there are also physiologic mechanisms where increasing heat, and this is actually part of the treatment planning, increased heat or elevating the temperature in a tumor will increase perfusion and increased oxygenation. And so this does also have an effect on enhancing radiation therapy. And then there is a more recent sort of attack that we've taken, and that is to explore areas where we induce immunogenic cell death through the magnetic hyperthermia in combinations with radiation. And I'll show a few slides on colorectal models that we've explored that in mice. So if one accepts that there is a motivation for using hyperthermia in the context of radiation therapy for recurrent or persistent cancers and tumors, then one can actually accept a little bit the logic that magnetic nanoparticle hyperthermia has advantages, because we really can take advantage of the fluidic nature of the heat source. And that means essentially that because the nanoparticles are suspended in a carrier fluid, they can be injected into the tissue, and therefore the heat source is from within the tumor. And the lost power, or the heat that is generated by the nanoparticles, can be adjusted remotely by adjusting the amplitude of the magnetic field. So activating and also controlling the amount of heating, or the thermal dose, if you will, can be done remotely. And so these are really sort of significant advantages over other technologies for delivering heat to tissues. And then, of course, there is this other aspect that we take advantage of, and that is that the thermoregulatory response is plastic and dynamic, and that is something that we deliberately want to put into our treatment planning scenarios. There is this other area that I've just put as a bullet point, and that is that iron oxide nanoparticles as an immune therapy, cancer immune therapy, is really an interesting new area of research in my laboratory. And basically, the short answer is that what we find is that doses, fairly high doses, systemically delivered systemic exposure to iron oxide nanoparticles actually induces pro-inflammatory processes, pro-inflammatory immune processes, and seems to reverse some of those suppressive, immunosuppressive aspects of tumors, of cancer tumors, that then initiates eventually T cell activation to suppress tumor growth. And what we see in transgenic models, spontaneously metastasizing transgenic models, breast cancer and lung cancer, non-small cell lung cancer, we see that a single exposure to the iron oxide nanoparticles essentially suppresses the formation of metastases quite significantly. And so this is actually an interesting area of research for us, but won't talk about that more than what I've done here. So as Costas mentioned, we have done a fair amount of work in my lab, and over the years I've done a significant amount of work as part of a collaboration with scientists at the National Institutes of Standards and Technology to really develop nanoparticles that can produce a heating profile that is tailored to what is useful from a clinical perspective. And really the significant piece in this graph is not just that the particles heat, but that they actually can generate the heat at low power or low amplitude magnetic fields. And part of the reason is, and we won't get into this, but essentially the consequent effect of exposing large volumes of tissue to an alternating magnetic field, depending on frequency and amplitude, causes non-specific heating through Faraday's law of induction, induced eddy current. And so what we want to do is we want to minimize the non-specific heating caused by the interaction of the magnetic field with tissue, and really focus the heating on what is generated by the nanoparticles. And just really a little bit of a sort of a pictorial primer on the origin of the heating and the frequencies that we use. The magnetic fluid hyperthermia, or the heating that we take advantage of, really comes about through specific internal magnetic processes in the nanoparticles. It's a process called hysteresis, magnetic hysteresis. And some of you might remember from your childhood days of playing with magnets. I did this when I was a kid. I would put iron nails, metallic nails, or any sort of ferromagnetic material onto a magnet, let it sit overnight, and then in the morning, the nail that was not magnetic the day before was actually magnetic. And so I could go around the house picking up other magnetic or other metallic objects with that. And that's really a property called remnants. And that is essentially pictorially represented on the left side here, which is if I take a virgin ferromagnetic material that has zero magnetization and put it into the presence of a magnetic field, the magnetization will rise to saturation depending on the applied field, which is H. And if the material exhibits this ferromagnetic property, then when I remove the magnetic field, so in other words, when H goes back to zero, there is a remnant magnetization that's defined by M sub R. And so what happens is if I take my external magnetic field and oscillate that magnetic field back and forth, I create this loop. And this loop is a hysteresis loop. And by forcing the magnets to traverse this hysteresis loop, depending on the area of loop, I actually generate heat. And I can generate substantial amount of heat, even though the individual loops themselves don't give off a lot of energy. We do this about 300, 400 times a second, 400,000 times a second, or 150,000 times per second. And so now you can see that a collection of these magnetic materials, as you see on the right, can substantially have an additive effect, each of these being potentially a heat source. And if I'm oscillating my magnetic field a couple 100,000 times per second, I can actually have quite a substantial effect. And then there is this, and I won't get into this in a lot of detail, but we've published on this material before. There is really a lot of nuanced physics on the magnetic field and the magnetic anisotropy that can be tuned or modulated depending on the particular structure, the crystalline structure of the nanoparticles, so that we can modify that heating response. And basically the particles that Costas talked about are the ones in the middle in panel B. Those are the JHU nanoparticles, and they exhibit a significantly different heating profile than the other two types of particles. And it's because of the internal magnetic structure that we deliberately engineered to take advantage of intra particle associations, magnetic associations. And so with that, that's about all I'm going to really talk about on the physics. So coming back to the motivation for the magnetic fluid hyperthermia, which is actually also interlinked with the motivation for the magnetic particle imaging. And this is really going to be talking about the translational aspects of my talk. And that is that while, as Costas mentioned, while magnetic nanoparticle or magnetic fluid hyperthermia has really been a clinical modality since about 2010, and it's because it offers some advantages, but there are some significant technological gaps that need to be overcome. One of these is that there is a lack of precise imaging and control of the nanoparticle activation, which is really a consequence of really the magnetic properties of the particles. And so there is a need for developing a next generation technology to directly image the nanoparticles, MRI and CT. It is well known that the iron oxide nanoparticles are a very strong contrast agent for MRI, but the significant challenge here is that the magnetic moments of the nanoparticles create such a strong effect that very low concentrations of the nanoparticles are required as a contrast agent. And these concentrations are too low for the magnetic hyperthermia. When we get to the concentrations that we need for the hyperthermia, essentially the susceptibility artifacts and many of the other aspects of the magnetic nanoparticles essentially make it impossible to use MRI for imaging the nanoparticles precisely in that context. And they're not significantly strong enough CT contrast agents, so cone beam CT doesn't work very well. So there is really a need to develop a new technology that can more precisely image the nanoparticles. A second part that is actually a challenge is that it is not possible. The physics are incompatible for MRI with the magnetic fluid hyperthermia. So one cannot actually do the heating, the magnetic fluid hyperthermia, or cannot do the heating in an MRI machine. And that's a subject for a different conversation. So the key challenges with nanoparticle heating arise from essentially one of the advantages, which is that the energy and the temperature control come from the nanoparticles, but the nanoparticle distribution in tumors tends to be heterogeneous. So therefore the heat generation tends to also be heterogeneous, and you could also have off-target tissue heating. As I was talking about the standard imaging technologies do not image the iron oxide nanoparticles directly. And then we also currently lack in situ and volumetric thermometry, so that we now have limits on the precision of our thermal dosimetry. And so these are actually all aspects of the technology development that Costas and I are working on, is explicitly part of the grant that we have to make improvements and to do some innovation around that. And there is this, I mean, even though it's not, I have no data and it's not a part of the talk, magnetic particle imaging is a newer technology, and it does actually offer the potential to advance magnetic fluid hyperthermia by providing the needed technology to do the quality assurance for the clinical treatments. And this is a subject that Costas and I talk about. All right, so I'm going to switch gears now to show you some data from our mouse models and different mouse models. As Costas has already shown you work from GBM, and as part of the collaboration, we are working with Costas on GBM. I've done some work in many other different models. So this is work that we did a few years ago, and this is really the punchline of this slide, is to really demonstrate to you the challenge in a very sort of real way, the challenge of the nanoparticle distribution once it's delivered into tumors. So in all of these cases, there are 3 different cases. These are all human prostate cancer cell lines that were grown in nude mice. So these are tumors sections taken, tumor xenograft sections taken from the mice. And the blue represents the iron oxide nanoparticles, the distribution of these particles a few hours after injection. This is Prussian blue stained tissue. And what you can see is that even though the nanoparticles were injected approximately in the center of the tumor, the nanoparticle distribution ends up being something very different. Now, the note here is we did not use CED. So Costas has been developing the convection enhanced delivery technique for many years. And that is really a far superior technique for delivering the nanoparticles. But even with that, the nanoparticle distribution after injection is going to really depend very much on the physical properties of the tumor itself. So the physical and physiological properties of the tumor. And you can see LIPC4 tumors down at the bottom have a lot of these interstitial spaces, and really that allows for a lot of the nanoparticles to distribute. So as much, and we'll see this in some of the simulations, you can basically surmise that this is close to the ideal, but we can't expect that. The other part that I want to highlight here is on the right. One of the things that we do in my laboratory is we will do inductively coupled plasma mass spectrometry to quantitate the iron that we in tissues. And essentially the result of this study is to demonstrate that even after injecting the same amount of material into the tumors, there is a significantly variable quantity of iron that is recovered. So within a few hours, what we find is that the nanoparticles not only distribute within the tumor, but there is some percentage of the nanoparticles in the fluid that will leave the tumor. And this is really actually a twofold problem. One is that now we have a reduction in the potency of our heating inside the tumor. And number 2, we now have the potential to overtreat or heat outside the tumor in areas that are not necessarily outside the target. And for tissues such as in brain, this can be a particularly challenging problem, because obviously one wants to, at the very least, control or minimize the external or outside target heating. So one of the things that we did, and this is part of our treatment planning. So the next few slides, what I'm going to do is really get into some of the mechanics on doing the heat transfer simulations as part of our treatment planning protocol that we're developing. But basically, conceptually, it works something like this, much like what you would have with radiation therapy planning. Number one, we would begin with imaging the target, and then also, assuming we have reasonable technology, getting an image of the nanoparticles and nanoparticle distribution. So the previous slide and this slide that I'm showing you now is essentially sort of the beginning point of assuming we have a known distribution of particles. And then the second piece is we would do the heat transfer simulations to essentially input the heat output of the nanoparticles, and then using the Penna's bioheat equation, we could then simulate or predict what kind of heat distribution or temperature distribution we're going to have in the target. And then this allows us to simulate various scenarios of amplitude and power modulation so that we can control the heating. And this assumes that we have some kind of temperature feedback control. So at least hopefully conceptually, you sort of got the picture. So right now what we're taking here is on the top row, we have digitized those histology images so that now what you see is essentially a sort of a, if you will, a binary image, black versus white, of the nanoparticle distribution, where each pixel would represent, each dark pixel represents a point heat source. And then down on the bottom row, what we've done for comparison is these are computer generated various simulated kinds of distributions for comparison. So one on the left, the uniform, that is, in some sense, that's considered to be the ideal distribution of nanoparticles. But one of the things that consequences that comes out from our simulations is that that's actually not necessarily what you want. There's the concentrated, where we just have all of the nanoparticles concentrated in the center of our simulated tumor. And then there is the Gaussian, which is this nanoparticles are concentrated in the center, but they're not uniformly distributed. It's a Gaussian distribution. In all of these simulated tumors have exactly the same number of heat sources. And so now when we go ahead and do the simulations, I think you can really see here pictorially what is a consequence or a potential consequence of these different distributions of the nanoparticles. And obviously, the more red represents a higher temperature. And as we go to cooler temperatures, we get to more blue. And you can see that on the scale on the right. And in this particular case, what we have done is we have inputted a constant power. So this is constant amplitude or constant power heating. And what you see is if you allow this to continue, basically you see a heterogeneous distribution of temperatures across the tumor, which depends very much on the concentration or the distribution of the nanoparticles. One of the consequences that is really worth keeping in mind here is that when we have the nanoparticles concentrated in a location, they can tend to generate more of a concentrated heating. And now the heat transfer properties sort of can't keep up. And so you get this kind of a potential runaway heating effect, which can lead to ablation inside that zone, which may or may not be desirable. But then the heat transfers away from that region. So if you look at the DU 145 case up top, you see that this particular effect is particularly pronounced. And because the concentration of the nanoparticles is off center and close to the tumor tissue boundary, what you see is that heat transfer bleeds into the normal tissue, and you're actually generating much more damage or off-target heating in the normal tissue. And that's rather an undesirable scenario. The LAPC4, as you can see down at the bottom, kind of represents more or less the idealized case where we get some heating around the target. As Costas talked about, you've got this invasive nature of GBM, and other gliomas have a very invasive nature. And so to some extent, having a modest heating or modest heat rise outside or in a margin would actually be desirable. an interesting feature of the uniform heating is actually that we have a higher rate of heat transfer across the tumor normal tissue boundary in the case of the uniform than we do in any of the other cases. And this is really just a consequence of the physics. So one of the aspects that we basically really have worked on to try to understand better, and Costas showed this a little bit. So the slide that I just showed you was if we applied a constant magnetic field, you see that the heating just basically goes up. Now we said, okay, well, what if we modulate the heating so that we control the power that's deposited and in order to control the temperature? Now the question really becomes, what's the feedback? What are you going to use for controlling the power? And so temperature feedback control is really what one would use in this case. And so in this particular case, the question that we asked is, given that we don't have in situ volumetric thermometry, what are we going to do if we have only a single temperature probe? Where can we put that temperature probe, and how do we modulate the power using that feedback control? And so this is really just a series of pictures that show what are really kind of consequences and how significantly different one can actually get temperature distributions based on putting the temperature probe at the center versus at the tumor tissue boundary. And what you see is that if I have the temperature probe in the center of the tumor, I really end up with undertreating the tumor, because basically the temperature is always shut down before it can build up enough heat. Whereas now if I put the temperature probe at the tumor tissue boundary, I allow enough heat to be generated by the nanoparticles in the center of the tumor so that there is a buildup of heating and the heat transfers out to the tumor tissue boundary, but I begin to shut it down before I get to the point where I overtreat. And so if we follow that sort of a scenario, what you can see is, and this is basically going back to the earlier slide that I showed you on those prostate cancer tumor models. On the left-hand side, we have constant magnetic field power, and basically what we end up is with a variable thermal dose. Whereas on the right-hand, so that's the constant magnetic field. On the right-hand side, we implement a variable heating power. And what you see, the red curve, the red trace, is actually the temperature probe that's at the tumor tissue boundary. It measures a much better, much more controlled heating profile, thermal dose. And you have in the other traces, these are temperature traces from other locations on the mouse, where we always measure the rectal temperature because we want to make sure that we don't overheat the mouse. We're measuring the contralateral skin location. And so you see, as Costas showed you, we can get a very nice separation between the tumor temperature, intratumor temperature, and the rest of the mouse. So that when we do the controlled heating as a radiation sensitizer, what we find, and these are Kaplan-Meier plots, what we find is that we have a much better effect on sensitizing the radiation treatment. If you look at the LAPC4, the control mice, and you can see that they're basically about 25, 27 days was the survival time of the entire group. And then if we do hyperthermia versus 5-gray radiation, we see that we improve survival a little bit or improve the control on the tumor, but that it's fairly modest, and that both of these give comparable results and extend survival to maybe 35 to 40 days. But if we combine the 5-gray heating with what we see is that we actually get a sensitizing effect that is even better than a higher dose of radiation. And that's really what we're after here. PC3 is both heat and radio resistant. So even in this particular case, we do see a beneficial effect of combining the hyperthermia with the 5-gray radiation, although it's not quite as good as 8-gray. And I mean, we actually studied this in vitro. We do see that the PC3 cells biologically are just inherently more resistant to both radiation and heat, which is why you don't see much of an effect on either the hyperthermia alone or even the 5-gray radiation by itself. But when they're combined, we do get a significant improvement. And that's really what we're after here is by combining the heat therapy with the radiation, we can essentially get a much more pronounced effect and much better response without actually having to go to the higher doses. And with that, I mean, I could stop now in the interest of time, but if people are interested, I could go on a little bit more to talk about these abscopal effects that we are able to pursue. It's not necessarily as relevant for GBM, but it is really something that is interesting. I'll leave it to you to let me know how you wish for me to proceed.
We've got at least 12 minutes left, so you can go longer as long as you leave some time for questions.
Okay, I will go through this fairly quickly. I mean, I will make the assumption that given that it's been talked about such a great deal, that there is a fairly general sort of common knowledge in the clinical community among your group on what we're talking about when we're talking about abscopal effects, and that is that we're basically inducing a treatment response away from treated site when we're doing local treatments such as radiation. And in this particular case, I will show, this is CT26. So these are tumor growth data from a subcutaneous colorectal model. It's a murine colorectal model. So CT26 are mouse-derived cells. And obviously one needs to do mouse-derived models here because one has to use fully immune intact animals. Whenever we're doing such immune-related studies, at least for the general experiments, for the initial experiments, we want to use it completely immune intact mice so that we can see that whether there is an effect or not. And so basically here we are, excuse me, what I'm showing you are tumor growth data of mice that were treated essentially by these different combinations. Radiation therapy was 5-gray. We had our hyperthermia, much like what I already showed you from the prostate cancer model. And then we have the combination. And you can see that basically the no treat control, the tumors grow much faster than they do for any of the treated groups. But we also see that, as we saw with the prostate cancer model, that when we combine the hyperthermia with the radiation therapy, we get a much better effect. Now, in this particular model, we had one tumor growing. We implanted one tumor on the thigh of the one mouse, and a mouse had implanted two tumors. One was on one thigh, the other on the other thigh. So essentially a bilateral tumor model. In all cases, mice were treated locally on one tumor. So let's say, for example, that we treated the tumor on the right-hand side. And what you're seeing here are the responses of the treated tumor. So we'll call that the primary tumor. We also measured the response in the distant tumor. So the tumor on the other side that was not treated. And what we see is that those tumors also responded quite significantly. They in some sense, in a very modest way, they did actually mirror the treatment combinations. In other words, the primary tumors showed a better response with the combination of hyperthermia with the radiation. And we see that the distant tumors showed a similar response. Now, these tumors, again, were not treated directly. So it does beg the question whether the treatment combination can induce an immune response that is either more robust or that includes more than one pathway so that there is a more significant immune pressure put on the tumor. And so that's something that we pursued in another model. And this is a much more challenging model. And it was a much more challenging set of experiments, in which case we combined radiation therapy, local, with the local hyperthermia plus immune checkpoint inhibitors. And the model here is the 4T1, which is a murine-derived triple negative breast cancer model, spontaneously metastasizes to the lungs. It's an extremely aggressive model. And what we find here is that the treatment effects always produce a response in the tumor. And we have the controls, which is no treat control, and then also surgically resected control. And you see that in this case, the tumors always regrow. Even when they're surgically resected, there is a residual number of cells that will regrow. So this is, in some sense, a really nice model to use for many sort of clinically relevant, challenging scenarios, because the tumor will return. So we have our single treatments. We have a high and a low dose hyperthermia. We have immunotherapy, which is the blue by itself. We have radiation therapy. In this case, radiation therapy is 3 times 8 gray, so 3 fractions of 8 gray. And then we have our dual treatments, which is the combinations of hyperthermia with immunotherapy. Never really as good as the radiation combinations with either hyperthermia or immunotherapy. And then the triple combination, which actually gives us really the most pronounced response. When we look at CD3 positive T cell infiltration after treatment into the primary tumors, and this is probably I would say, really one of the main punchlines that we learned from this study, and that is that the radiation and radiation plus hyperthermia and or immunotherapy, but radiation is actually probably the most significantly immune activating or immunogenic therapy, in the sense that it really induces a great deal of infiltration into the primary tumors after treatment. And the response of the treated tumors to the radiation really does depend a great deal on the immune response, because when we inhibited T cells using pharmacologic inhibition, what we found was that our treatment response on the primary tumor was diminished. And this actually, I think, is quite significant, and probably even really important for GBM, in the sense that I think it is important to combine more than one therapy, if for no other reason, because one is able to induce damage into the tumor tissue and thereby initiate responses in the tumor that will activate will induce immune activation. When we looked at the metastases, we do see a modest reduction in the numbers of metastases following treatment, and there's all these different combinations. I would encourage you to read the paper in the interest of time. And we do see that the triple treatment on the far right actually gave us a little bit more of an effect on the tumor metastases compared to even the surgically resected controls, but it wasn't really so significant in the sense that we are seeing something that's overwhelming. And so there is a significant effect on just cytoreduction or removal of the tumor as a source of metastases. We do see in the lungs of these mice that the T cell infiltration is increased, but the aggressive nature of the tumors, and there's quite a bit of interesting immunologic features about the BALB/c mice that does sort of predispose them to having a weaker immune response. But even with this, we see that there are some encouraging aspects of this. One of the lessons that we really did learn is that we really do need to get much better control of the hyperthermia, which is really the point of the entire project that Costas and I talked about. There's the Kaplan-Meier curve. So we do see that the best sort of survival enhancing treatment combination was the radiation with immunotherapy. And with that, I think I will conclude.
Thank you both for presenting today. This is a fascinating combination of talks with a lot of really important clinical implications. I did have one question for both of you, which is when you have, when you're assessing these tumors that we would consider nanoparticle convection enhanced delivery for, is there a good way without pathology to potentially assess the likelihood of good distribution within the tumor, perhaps with newer imaging techniques to assess that in advance?
Yeah, I'll say my thoughts on that, and then maybe Rob can add. I think, Peter, there's been some studies done looking at the hydrostatic pressure of tumors, and it's actually quite intriguing, because while we think it's easy to get things into tumor if you're injecting, it's actually not, because the tumor pressure pushes the agent out. So that's why convection enhanced delivery is a better alternative to stereotactic injection, because you're providing a continuous pressure gradient to really perfuse the interstitial spaces of the brain. But even with that, there are some challenges. So imaging, is something that we use to look at the tumor, and certainly the necrotic portion of the tumor, may be an area where we can get into. It's a softer, more liquid portion of the tumor, but the challenge is really the periphery, and that's where I think we need to get into the surrounding brain as well. And that's where some of the fine tuning that Rob was talking about is, how do you heat that margin without hurting the surrounding brain and target those cells? And that's where the moderate hyperthermia, I think, becomes really important.
I had a similar question if there's time. Hey, Dr. Pev, this is Alex Schuper, one of the junior residents. Thank you so much for your time. It was a really interesting talk. Kind of along the same lines of Dr. Morgenstern, Dr. H and I earlier this week, we were talking about how there's certain heat sinks in the context of LITT. And obviously it's a similar type of thermal induction. I'm wondering, do we see the same thing in magnetic therapy in terms of certain types of vascularity within the tumor? And if we have any evidence as far as receptors, we know in the GBM population, certain areas of the tumor are going to overexpress receptors like EGFR more. Do we see any relationship between the types of magnetic responses in these certain populations of overexpressed receptors or vascular proliferation like we do, and a similar response that we see in therapies like LITT?
Costas, did you want to?
No, why don't you jump on that, Rob?
I'll speak more from the physics and the aspect of the question that you're getting to, these heat sinks. So the answer is yes, we do know of these effects. And so basically the strategy or the idea is to conceptually, if one has sufficient thermometry, then the treatment algorithm would basically be able to take advantage of those nonlinearities, if you will, in the response. Because in some places where you have less of a heat sink effect, you're going to get more of a runaway heating or an increase there, an accentuation of the heating, and that offsets the heat sink effect in the other areas. All right, it's a really sort of a very complex question that you're asking, and it's a really sort of a complex answer. But what I'm going to basically respond with is this. It's actually part of our strategy to deliberately exploit the nonlinearities on physiologic response to heat. That is the heat sink effect, not the nonlinear temperature response on perfusion as well, because as you heat mildly, actually perfusion increases because you get vasodilation. And so that gives you a heat sink effect. But when you go to a certain point and start to damage the tissue in that area, then you get a vascular collapse. And now you actually have a sort of an insulating effect, and you start to get more runaway heating. And so that's why we're basically going to doing these more complex heat transfer simulations, because as you saw on the graphs that we showed, the response of the heating response of the nanoparticles is nonlinear with the magnetic field amplitude. So if we're in the right range, we can just do a slight increase in the magnetic field amplitude. We get a big boost in the heating. And so what we want to do with that is, conceptually, you basically take advantage of the fact that if you might have a heat sink, a strong heat sink effect, you do higher rate of heating here, you get collapse of those tissues locally, so that now you basically shut down, to some extent, you might shut down that heat sink effect or reduce it. So now that you allow that heat transfer to go on. But you don't want to go so far that you overtreat, and now you start to damage the tissue in the surrounding normal tissues. So to know whether what you have beforehand, I don't think there is a technology that exists yet that would allow us to do that. I mean, there's perfusion MRI, there's DCE MRI, all of these kinds of different technologies that might be beneficial, but I don't think we really know enough to be able to see how we utilize them. And sort of taking a practical engineering approach, I would say, well, I would rather be able to image the nanoparticles that I already have, and then be able to measure the temperature response function in the tumor as I'm doing the heating, and then have my treatment algorithm make the adjustments based on this complex sort of scenario. That's really kind of the approach that we're taking with this. It's really a much more pragmatic engineering approach, and say, if we have all of the data, if the technology exists to provide us with the data, in other words, we're getting the temperature feedback, we have imaging of the nanoparticles after we've injected them, we can allow the treatment algorithm to compensate for all of these other scenarios. Hope that answers the question.
Yes, thank you so much.
I think Joanna had a question. I'll just tell everyone the question. So Joanna is asking whether there's a relationship between the intensity of hyperthermia and post-LITT side effects, specifically brain edema, and how do we best treat that, since steroids may suppress the immune response? So that's actually a very good question, Joanna. So part of how we assess edema after hyperthermia is based on the volume that we're treating. It's almost similar to the volume we treat with radiation. We know that if we treat a large volume brain tumor with high doses of radiation, we're going to see surrounding edema and radiation effect that can ensue. And then we give steroids, and sometimes we take those patients even to the OR to decompress the brain. But in hyperthermia, there's a similar volume relationship with that type of edema. And certainly the higher the heat or the ablative type approach, which we do with LITT, can cause what Rob said is really destruction of the blood vessels. And then you have kind of a more insulated effect, but then you also ensue an inflammatory response, which causes some of the edema. So we're really looking at some of a more moderate hyperthermia approach, where we really don't want to blast the brain with a high temperature, but really kind of target the cancer cells in that sweet spot. That's what we're trying to figure out, that sweet spot where we can get the cancer cells, but keep the surrounding astrocytes and neurons still working well, and minimize any toxicity.
Yeah, if I might just add to that, and recognizing completely that I'm not the clinician here, but from some of the data that we have collected in our mouse models, including the transgenic models, it really, what it comes down to is being able to have your intervention be modulated so that you initiate the right level of a response. Because an immune response and an activation of an immune response, we see it can be very beneficial in the context of cancer and in helping with the treatment response in the tumors. But it can also go the other way, and particularly in sensitive tissues like in brain, you don't want a runaway inflammatory response that then induces all kinds of additional damage. So it's really about modulating on a case-by-case basis and having that really fine control, which is what this project is all about, actually.
Yeah. Great. Well, I think we ran out of time. Rob, once again, thank you for joining us, and I'm really happy we were able to share our work. It's always exciting, and I think we're getting on a call in 5 minutes, right?
Yeah, we are.
So-
Yes, project call on this exactly. Yeah.
Yeah.
Well, thank you
Yeah.
very much for the opportunity and the invitation to speak with you. I hope it was informative.
Awesome. Thank you.